15% of the human population is walking around with blood missing something almost every other person on Earth carries. Not a disease, not a disorder. A protein simply absent, coding none of their red blood cells, an absence written into their DNA and passed down for thousands of years. 85% of humanity carries that protein without exception. A marker so ancient it runs unbroken through the entire primate family tree, chimpanzees, gorillas, every close relative humans have ever shared a branch with. The remaining 15% do not have it, and for decades, doctors could describe exactly what was missing without being able to explain, in real time, why it was capable of turning a routine transfusion into a life-threatening event. That question first exploded into view in a delivery room, not a laboratory, not a research paper, but in the middle of an emergency where a woman's own blood suddenly turned against the one transfusion that was supposed to save her life. A young mother lies in a delivery room in New Jersey in 1937. Her baby has just been born dead. She has lost a dangerous amount of blood during the delivery, and doctors need to act fast. Her husband is the same blood type. Oh, a perfect match. By every rule medicine understood at the time, they transfuse his blood into her veins to save her life. Within minutes, her body turns against it. Chills, fever, her own blood attacking the very blood that should have saved her. By every existing law of transfusion science, this should not be happening. Same blood type. Same donor pool that had worked 1,000 times before. And yet her immune system is treating her husband's blood as an invader, as something foreign, something that does not belong. The doctors in the room have no explanation. Nothing in the textbooks accounts for what they are watching happen in real time. That single unexplainable reaction in that one hospital room was about to unravel a hidden layer of human biology that no one had ever detected before, one that would eventually be found in the veins of hundreds of millions of people alive today, one tied to an ancient European population, a dramatic medical rescue decades later, and a story that is still being refined by scientists today. To understand how strange this reaction really was, you have to remember how confident medicine already felt about blood by 1937. Nearly 4 decades earlier, in 1900, an Austrian physician named Karl Landsteiner had discovered the ABO blood group system, the classification of blood into types A, B, AB, and O that still governs every transfusion performed today. By the 1920s, ABO testing had become routine in hospitals across the world, and doctors believed that matching a patient's ABO type to a donor's was the entire safety check required. A husband and wife sharing the same ABO type, as this couple did, should have been about as safe a transfusion as medicine could offer. That is exactly what made the reaction in that delivery room so alarming. That case belonged to physician Philip Levine, working alongside his colleague Rufus Stetson at Newark Beth Israel Hospital, when they tested the mother's blood serum against blood from other donors who were supposedly compatible. The results were staggering. Her serum attacked the red blood cells of 80 out of 104 people who were, on paper, a perfect ABO match. Something else was going on beneath the surface of human blood, something the ABO system discovered decades earlier by Karl Landsteiner had never accounted for. Levine and Stetson published their findings in the Journal of the American Medical Association in 1939, describing what they called an unusual case of intragroup agglutination. It was a modest title for a discovery that would eventually reshape obstetric medicine, transfusion protocols, and the way scientists understood the genetic diversity hidden inside a substance most people assumed had already been fully mapped. They didn't have a name for the culprit yet. They just knew it was there, hiding in plain sight inside blood that scientists thought they already understood completely. The answer arrived the following year in 1940, when Karl Landsteiner, the same man who had already reshaped the field of hematology, teamed up with Alexander Wiener. They injected blood from a Rhesus macaque monkey into rabbits and guinea pigs. The animals' immune systems produced antibodies in response. And when researchers tested those antibodies against human blood samples, something remarkable happened. The antibodies reacted with 85% of the human blood tested. It attacked the blood cells of the overwhelming majority of people, but left a smaller group completely untouched. They had found the antigen. They named it the Rhesus factor, after the monkey whose blood had first revealed it. It fit precisely with what Levine and Stetson had already found in that delivery room. The mother's blood was missing it entirely. Her husband's blood carried it. That invisible difference was enough to turn a life-saving transfusion into a medical emergency. Once scientists understood what they were looking at, the deeper mystery of Rh-negative blood is that it is not a variation of Rh-positive blood. It is a complete absence. Every red blood cell in the human body is coated with dozens of surface proteins that act like identification markers, telling the immune system what belongs and what does not. The Rh factor, more specifically what researchers call the D antigen, is one of the most significant of those markers. If you carry it, your blood is Rh positive. If you do not, your blood is Rh negative, and your immune system has never learned to recognize that protein as part of you. Roughly 85% of people worldwide carry the D antigen. The remaining 15% simply do not have it. Not a weaker version, not a damaged version. A total absence of a protein that almost everyone else on the planet carries on the surface of every red blood cell in their body. For decades, that 15% puzzled researchers, not because the biology was mysterious in the way internet legends now claim, but because tracking the actual genetic mechanism behind it required tools that simply did not exist yet. It took until 1982 for scientists to first identify the Rh proteins themselves at the molecular level, unusually hydrophobic proteins sitting in the red blood cell membrane carrying no sugar chains at all, which is strange for proteins on a cell surface. And it took even longer into the 1990s and 2000s for geneticists to fully map the gene responsible. What they eventually found was not some untraceable void in the genetic record. It was a gene called RhD, sitting on chromosome 1, right next to a closely related gene called RhCE. In the majority of Rh-negative Europeans, the RhD gene is not mutated or corrupted. It is simply missing, deleted entirely from that stretch of DNA, likely the result of an unequal crossover event between RhD and its neighboring gene sometime in ancient human history. No D antigen gene, no D antigen protein, no D marker on the blood cell surface. In other populations, particularly people of African and Asian descent who carry Rh-negative blood, the mechanism is different again. The gene is sometimes still present but silenced, switched off through separate mutations that block it from producing a working protein. Rh-negative blood, in other words, did not spring from nowhere. It came from a well-documented category of genetic event, a deletion, that geneticists observe elsewhere throughout the human genome and throughout the animal kingdom. The Rh system itself turned out to be far more complicated than that first single D antigen ever suggested. Researchers have since identified more than 50 distinct antigens within the Rh blood group system, encoded across the two closely linked genes, RhD and RhCE, which sit side by side on chromosome 1 and occasionally swap segments of DNA with each other in a process called gene conversion. That constant genetic shuffling produces the dizzying variety of Rh subtypes seen across different populations, including rare partial D variants where a person carries only a fragment of the D antigen, sometimes causing confusing results on standard blood typing tests. Even after scientists located the gene, one question lingered for years afterward with no clean answer: what these proteins are actually for. The leading theory is that Rh proteins help transport ammonium and carbon dioxide across the red blood cell membrane and may help maintain the physical stability of the cell itself. In other words, the same protein responsible for one of the most dramatic stories in transfusion history may, at its core, simply be a piece of cellular plumbing. What makes this genuinely fascinating is not that science cannot explain where it came from. It is where this variant is concentrated and how unevenly it is spread across the human species. If Rh-negative blood were the result of a mutation that happened randomly and repeatedly across scattered populations, you would expect to see it spread out fairly evenly wherever humans settled. That is not remotely what the data shows. In most of Europe, roughly 15 to 16% of the population is Rh-negative. In parts of sub-Saharan Africa, that number drops below 1%. In East Asia, the frequency falls to somewhere around 0.1 to 0.4%, vanishingly rare. Among indigenous populations across the Americas prior to European contact, Rh-negative blood was close to non-existent, a detail that lines up with the timeline of human migration into the continent many thousands of years before the deletion became common in Western Eurasian populations. This is not noise. This is the fingerprint of a specific, ancient genetic event that occurred in one lineage, in one place, and then traveled outward with that lineage's descendants, rather than appearing independently around the globe. And nowhere does that fingerprint show up more dramatically than among the Basque people, an ethnic group whose homeland straddles the western Pyrenees mountains, spanning parts of northern Spain and southwestern France. Multiple population studies have recorded Rh-negative frequencies among Basques, reaching into the 30 to 35% range, roughly double the average across the rest of Europe, and by some genetic estimates of the underlying deletion allele itself, even higher than that. The Basques are also famous for something else entirely unrelated to blood, and yet strangely connected in the bigger picture. Their language, Euskara, has no confirmed relationship to any other language spoken on Earth, living or dead. It is what linguists call a language isolate, untouched by the wave of Indo-European languages that swept across the rest of the continent thousands of years ago. Basque genetics tell a similar story of deep continuity, tested far more precisely in recent years through whole genome sequencing rather than the older, cruder blood group surveys of the mid-20th century. Studies of their DNA suggest a population that experienced comparatively little admixture with the migrating farming populations that reshaped the genetic map of Europe during the Neolithic period, roughly 7,000 to 8,000 years ago. In plain terms, The Basques appear to be one of the more direct genetic threads connecting modern Europeans back to some of the continent's earliest post-Ice Age inhabitants, a population that took refuge in the Franco-Cantabrian region during the depths of the last glacial period and never fully dispersed the way surrounding groups eventually did. Some geneticists trace that continuity back even further to the last glacial maximum roughly 20,000 years ago, when much of northern Europe was locked under ice sheets and human populations were pushed into a handful of survival refugees along the continent's southern edge. The region spanning what is now northern Spain and southwestern France, the Franco-Cantabrian refugium, is widely considered one of the most important of these Ice Age shelters, a place where small bands of hunter-gatherers weathered the coldest millennia of the Pleistocene while much of the rest of the continent was uninhabitable. Some researchers argue that a significant portion of Western Europe's post-Ice Age population growth traces back to descendants of exactly these refugee populations, radiating outward from the Pyrenees and the surrounding region. If that model is correct, the Basques may represent a population that never fully left, staying rooted close to the original refuge while their distant relatives spread across the continent and mixed extensively with newcomers over the following millennia, from Neolithic farmers arriving from Anatolia to the Bronze Age migrations that later swept through much of Europe. That isolation is very likely the real explanation for why Rh-negative blood concentrated so heavily in this one corner of Europe. When a population stays relatively closed off, marries largely within itself, and does not repeatedly mix with incoming groups, genetic traits that exist within that founding population, even ones that started out uncommon, can rise sharply in frequency over generations purely through founder effects and genetic drift, without needing any dramatic explanation beyond geography and time. The Pyrenees Mountains functioned almost like a genetic vault, sealing in traits that elsewhere across Europe were diluted as new populations arrived, intermarried, and spread their own genetic signatures across the continent. Elevated Rh-negative rates also show up, to a lesser degree, in Ireland, Scotland, England, parts of Scandinavia, and among Berber populations in the Atlas Mountains of North Africa and other communities across the Mediterranean world, all regions with their own histories of relative geographic isolation along the western edge of the continent. Now here is where the story stops being only about ancient migration and becomes something far more immediate and, for a long stretch of the 20th century, genuinely dangerous. Rh-negative blood does not just complicate transfusions. It complicates pregnancy in a way that shaped and limited its own spread for generations. When an Rh-negative mother is carrying an Rh-positive baby, having inherited that positive status from the father, a small amount of the baby's blood can cross into the mother's bloodstream, especially during delivery. Her immune system, encountering the D antigen for the first time, treats it as a foreign invader and begins producing antibodies against it. In a first pregnancy, this is rarely dangerous because the process of sensitization typically happens too late to affect that first baby. But the mother's immune system does not forget. In a second Rh-positive pregnancy, those antibodies are already present and ready. This time, they cross the placenta early and attack the fetus's red blood cells directly, causing a condition originally called erythroblastosis fetalis, now known as hemolytic disease of the fetus and newborn. The consequences could be severe anemia, dangerously enlarged organs, jaundice so extreme it caused permanent brain damage known as kernicterus, or death before or shortly after birth. Before doctors understood any of this, the condition had already been observed for centuries without explanation. A French midwife documented a case in a set of twins as early as 1609, one stillborn and hydropic, the other jaundiced and dead within days from kernicterus, with no one able to say why. For the next 330 years, doctors watched families lose children in this pattern with no understanding of the cause, often blaming difficult labors or simple bad fortune, because nothing in the visible symptoms pointed toward an invisible mismatch happening at the level of a single surface protein on a blood cell. Some families lost multiple children in a row without any doctor connecting the deaths, because the pattern only becomes visible in hindsight once you know a second or third pregnancy is more dangerous than the first, and why. Once Levine and Landsteiner's discoveries connected the dots in the early 1940s, the scale of the problem became horrifyingly clear. In the United States alone, prior to any effective treatment, Rh disease was killing an estimated 10,000 infants every single year, and leaving many more survivors with permanent neurological damage. Physicians developed the first real intervention in the 1940s. An exchange transfusion pioneered by Alexander Wiener himself and refined by Harry Wallerstein, in which a newborn's Rh-positive blood, already under attack from maternal antibodies, was slowly replaced with compatible donor blood. It was crude and risky and had to be performed immediately after birth on infants who were often already critically ill. Yet, over the 2 decades, it remained standard of care. That grim procedure is credited with saving roughly 200,000 lives. The real turning point did not come until the 1960s, and it came from 2 directions at once. In Liverpool, England, physicians Cyril Clarke and Ronald Finn began exploring a way to prevent the mother's immune system from ever becoming sensitized in the first place. At almost the same time in New York, at Columbia Presbyterian Medical Center, obstetrician Vincent Frieda and blood bank director John Gorman, working with immunologist William Pollack at Ortho Pharmaceuticals, pursued the identical goal from a different angle. Their theory was almost paradoxically simple. If you inject an Rh-negative mother with a carefully measured dose of anti-D antibody immediately after delivery, before her own immune system mounts its slower natural response, those injected antibodies quietly mop up any of the baby's Rh-positive cells that crossed into her bloodstream before her body recognizes them as a threat. No recognition, no sensitization, no antibodies waiting for the next pregnancy. Gorman tested the concept first on his own sister-in-law in January of 1964, a small, personal first step for treatment that would go on to reach millions of families worldwide. Clinical trials followed across more than 40 medical centers, spanning the United States, Britain, Germany, Sweden, Italy, and Australia. The results were unambiguous. Among treated mothers, the sensitization rate dropped from roughly 14% to about 1%. In 1968, the FDA approved the treatment for use in the United States, under the brand name that would soon become familiar to expectant mothers everywhere, Rho-Gam. A New Jersey mother named Marianne Cummins, pregnant with her third Rh-positive child and desperate to receive the injection before her due date, became the first patient to receive the approved treatment just hours after it reached the market that spring. Within a single generation, a disease that had been killing thousands of American infants every year and quietly narrowing Rh-negative family lines for as long as the trait had existed was reduced to a rare and largely preventable complication. It remains, more than 50 years later, one of the most cost-effective medical interventions ever developed, still administered routinely to Rh-negative mothers worldwide during pregnancy, and again shortly after delivery. Sitting inside the broader Rh-negative population is one subgroup that carries its own separate distinction and its own separate paradox. O-negative blood, found in somewhere between 5 and 7% of people globally depending on the population studied, is famous in emergency medicine as the universal donor. Because O-negative red blood cells carry neither A nor B surface antigens from the ABO system, nor the D antigen from the Rh system, they trigger no immune rejection in patients of virtually any blood type. When a trauma patient arrives at a hospital unconscious and bleeding, with no time to run compatibility tests and no medical history to consult, O-negative is what goes into their veins, because it is the one type doctors can trust to work regardless of who the patient turns out to be. It crosses every boundary the rest of the blood typing system creates, but the relationship runs in only one direction. People with O-negative blood can only safely receive transfusions from other O-negative donors. Every other blood type is incompatible with their immune system. It is a strange asymmetry, biologically speaking, a blood type that gives to everyone and can only take from a small fraction of the population in return. And it is precisely why blood banks worldwide chronically struggle to keep enough O-negative in reserve, since it is used constantly in emergencies, but can only be replenished by one of the rarer donor groups. The idea of the universal donor did not become practical medicine overnight. The infrastructure of modern blood banking was largely forged under the pressure of war. During the Second World War, physicians, including Charles Drew, helped pioneer large-scale blood plasma collection and storage programs, making it possible to stockpile blood and ship it to battlefield hospitals, rather than relying only on direct donor-to-patient transfusion. Those wartime techniques carried directly into civilian hospitals afterward, and it was in that post-war system that the practical value of O-negative blood became impossible to ignore. In trauma medicine, there is rarely time to type and cross-match a patient's blood before they bleed out. O-negative became the default reserve for exactly that reason, and today it remains the blood type most requested by trauma centers and ambulances worldwide, even though the people who can donate it make up such a small slice of the population. Beyond the hard genetics and the transfusion medicine, there is a cultural layer to this story that is worth being honest about, because it explains why Rh-negative blood keeps resurfacing online in far stranger forms than anything in a medical journal. Because Rh-negative blood is rare, geographically clustered, tied to an unusually old and isolated European population, and involved in one of medicine's more dramatic rescue stories, it has become fertile ground for speculation that goes well beyond what the science actually supports. Theories describing Rh-negative carriers as descending from some untraceable non-human ancestor, or possessing unusual psychic sensitivity, or elevated intelligence, none of which has any basis in peer-reviewed research. The real story does not need any of that embellishment to be remarkable. A stillbirth in a New Jersey hospital in 1937 led, within 30 years, to the identification of an entire blood group system, the unraveling of a genetic deletion carried by hundreds of millions of people. A clear demographic map connecting modern Rh-negative populations back to some of post-glacial Europe's most isolated survivors, and a medical breakthrough that has spared an untold number of families from losing a child to a condition that had no name for over 3 centuries. Ancient DNA research is still refining exactly when and where that original RhD deletion first appeared, with current estimates placing its origins in a small founding population somewhere in ancient Western Eurasia, well before the major agricultural migrations that reshaped the continent's genetic landscape roughly 7,000 to 8,000 years ago. Each new ancient genome recovered from Ice Age era remains adds another data point to that timeline, sharpening a picture that has taken more than 80 years and thousands of individual research papers to bring into focus. And just unlike the version of this story that circulates in comment sections and late-night internet forums, nothing about that picture requires abandoning known biology to make sense of it. A deletion on chromosome 1, inherited generation after generation through a population that stayed relatively isolated for thousands of years, is a mechanism modern genetics understands well, even if the exact moment it first occurred is still being pinned down more precisely with every new sample scientists manage to sequence. What began as a single unexplained transfusion reaction in a delivery room in 1937 turned out to be a doorway into one of the more intricate stories written into human DNA, a story about a small isolated population somewhere in ancient Europe, a genetic deletion that quietly rode along through their descendants for thousands of years, a reproductive complication that shaped which families could pass that bloodline forward, and a scientific breakthrough that finally broke that ancient constraint within a single generation. The blood running through roughly 1 in 7 people alive today carries a piece of that history in every red blood cell, an absence rather than a presence, a protein that simply was never there, tracing back not to any mystery science refuses to solve, but to one of the more remarkable and well-documented threads in the story of who modern humans actually are and where they came from. If your own blood type happens to be Rh negative, you are carrying a genetic signature shared by a relatively small and specific slice of humanity, one whose deepest roots may reach back further into Europe's ancient past than almost any other trait still common in the population today. What part of the world are you watching from? Drop it in the comments, and if this kind of deep dive into the science hiding inside your own biology is something you want more of, make sure you subscribe.